Before a bacterial cell divides, it must first make an exact copy of its DNA. This remarkable process ensures that each daughter cell receives a complete and identical set of genetic information. So, let's explore how bacterial DNA replication takes place step by step. The process begins at a specific location on the bacterial chromosome called the Origin of Replication, or OriC. The initiator protein, DNA, recognizes the origin sequence and binds to it, causing the DNA double helix to open. This marks the beginning of DNA replication. Next, DNA Helicase, also known as DNAB, unwinds the DNA by breaking the hydrogen bonds between complementary base pairs. As the two DNA strands separate, two replication forks are formed and move in opposite directions around the circular chromosome. At the same time, single-stranded binding proteins, or SSBs, attach to the separated DNA strands to keep them stable and prevent them from rejoining. Meanwhile, DNA gyrase, a type of topoisomerase, relieves the twisting tension. Once the DNA is fully unwound, the enzyme Primase, also called DNAG, synthesizes short RNA primers. These primers provide the free 3-hydroxyl end required for DNA polymerase to begin synthesizing new DNA strands. Now, DNA Polymerase III, the main replication enzyme in bacteria, starts adding complementary nucleotides in the 5-3 direction. Because DNA strands are antiparallel, replication occurs differently on each strand. The leading strand is synthesized continuously toward the replication fork. In contrast, the lagging strand is synthesized discontinuously away from the replication fork as short DNA segments known as Okazaki fragments. DNA Polymerase I removes the RNA primers and replaces them with DNA nucleotides. Finally, DNA ligase seals the remaining nicks between the Okazaki fragments, joining them into one continuous DNA strand and completing the lagging strand. Replication continues until the two replication forks meet at the termination region of the bacterial chromosome. Topoisomerase IV then separates the two newly replicated circular DNA molecules, ensuring that each daughter cell receives its own chromosome. The final result is two complete and identical DNA molecules, each composed of one original parental strand and one newly synthesized strand. This mechanism is known as semi-conservative DNA replication, and it ensures the accurate transmission of genetic information from one bacterial generation to the next.